Welcome everyone. Today we are going to learn about regulation of cytosolic pH by antiporters. Now the first question is why regulation of cytosolic pH is important. The answer is most proteins operate optimally at a particular pH. Like lysosomal enzymes work best at low pH, that means at pH of 5. And the cytosolic enzymes function best at close to the neutral pH, that means pH 7.2. So cells try to maintain the cytosolic pH. The cytosolic pH try to remain approximate 7.2, that means close to neutral pH. Now for this maintenance, 3 antiporters are dedicated. The first antiporter name is sodium hydrogen antiporter. The second antiporter is called chlorine bicarbonate antiporter. Now this chlorine bicarbonate antiporter is actually sodium dependent. Remember, this antiporter is sodium dependent. And another antiporter present, that is also chlorine and bicarbonate antiporter. But this antiporter is actually sodium independent. Sodium independent. Now, these two antiporter, the function of these two antiporter increases when the pH of the cytosol actually fall. That means the pH becomes less than 7 or 7.2, and that means that there is increase in hydrogen ions. So basically pH fall means acidity of the cytosol increases. That's why H plus ions get increases. So H plus ions increases. So what I have mentioned, that these two antiporter, sodium hydrogen antiporter and chlorine bicarbonate antiporter, that is sodium dependent, the activity of these two antiporter increases when the pH fall, that means fall below 7.2, that means acidity increases, that means hydrogen ions increases. But this antiporter works at its best when the pH actually rise above 7.2, that means it becomes too alkaline, and that means the hydrogen ions actually drops. So by this way, it actually maintains the pH at 7.2. So how it works, I will explain. Basically, in case of sodium hydrogen antiporter, sodium ions actually move inside the cell, and hydrogen ions are actually exported. So hydrogen actually export out. Now one thing we have to understand, that this sodium import is actually driven by 2 points, two factors. One is called chemical concentration gradient and membrane potential. Remember these 2 points. The driving force of sodium inside the cell depends on these 2 factors. These 2 factors actually acts as driving force. Remember the term driving force. And togetherly, this 2 factor is called electrochemical gradient. Electrochemical gradient. Basically, in case of sodium, the outside concentration of sodium is high, approximately 145 millimolar, and inside is 12 millimolar. So there is 12-fold change in the concentration of sodium in the exterior than inside, that means cytosols. So there is a chemical concentration gradient of sodium present. Not only that, outside of the membrane is positively charged and inside is negatively charged, and that's also try to that also drives the sodium to get inside in the cells, because positive ions are attracted towards the negative ions. So this is the driving force of sodium. So sodium actually goes down the concentration gradient. It goes from higher to lower concentration, and that movement actually helps or release the energy to move hydrogen inside to the outside. And by this way, what happens? As I have mentioned, this 2 antiporter works when the pH falls. That means the hydrogen ions increases, and by removing hydrogen ion from inside to the outside, it actually decreases the hydrogen concentrations. That means it increases the pH. So during the fall, it actually exports excess hydrogen ions outside the cell. This is the first antiporter that actually regulates cytosolic pH. Now, how chlorine bicarbonate antiporter, that is sodium dependent, works? Here also sodium actually transported inside the cell along with bicarbonate ion, and simultaneously chlorine is exported and one hydrogen ions actually export out. So as you can see, one positive ions import, one positive ions export, one negative ions imported, and one negative ions exported. But what happens here? See here, just like the sodium hydrogen antiporter, this antiporter also removes one hydrogen ions. That means it actually decreases the hydrogen concentration, H plus ions concentrations. But this antiporter actually works twice efficiently than this sodium hydrogen antiporter. So this antiporter works twice efficiently. Remember, twice efficiently. Now, how this transporter works twice efficiently? Because first is it removes one hydrogen directly from inside to the outside. Another is by importing bicarbonate ions, it actually causes, I want to write here, by importing one bicarbonate ions, it actually goes through one reaction, that is the bicarbonate when get inside in the cell, it actually converted by an enzyme called carbonic anhydrase, into carbon dioxide and OH minus, that means hydroxyl group. See, by importing one HCO3, this HCO3 is divided into carbon dioxide and hydroxyl ion. Now this carbon dioxide actually diffuses out of the cell, but this hydroxyl ion remains, that actually neutralizes the excess hydrogen ions that is present when the pH falls. Because I have mentioned, when pH falls, the hydrogen ions increases. Now these excess hydrogens are actually neutralized with the hydroxyl group to generate water. By this way, this antiporter remove one hydrogen by directly exporting out, and by importing one bicarbonate, it actually neutralizes another hydrogen ions. That means it can neutralize two hydrogen ions. So by this way, this antiporter works twice efficiently than sodium hydrogen antiporter. Now, how this chlorine and HCO3 antiporter works? As I have mentioned, this antiporter works in reverse of these two antiporter. Here, when the pH rise, that means it actually goes above the 7.2 pH, it becomes alkaline. That means the hydrogen ions here is low. Now this antiporter will try to increase the hydrogen ions. How it all works? See, this antiporter actually remove one HCO3 ions and import one chlorine ion. The reverse occur. Here, as you can see, chlorine is exported and bicarbonate is imported. Here, opposite occur. Now, why this HCO3 removing will increase the hydrogen concentration? Let me show you. Here, what happens during aerobic metabolism? Carbon dioxide is actually generated. Now, this carbon dioxide will react with the water present in the cytosols. that will make by H2CO3 this acids. Now this acid is actually a very weak acid. It readily dissociates into this H2CO3 readily dissociates into HCO3 minus and H plus ions. See, carbon dioxide reacts with water, makes H2CO3. This acid is weak, so it will dissociate into HCO3 and H plus. Now, if we remove this HCO3 from these cells, what will remain? One hydrogen ions will remain, and by this way, the hydrogen ions or that means the protons are actually increases inside the cell. So this antiporter don't need the help of sodium, but it actually increases the hydrogen ions by exporting the bicarbonate. This is the reactions that occurs, and this is the reaction how this antiporter actually lowers the pH, because here hydroxyl ions actually neutralize another hydrogen atom. So I think you have understood these three antiporters, how it actually maintains during the fall of the pH and during the rise of the pH. These three antiporters actually maintain the pH, and it actually maintains at about 7.2, that is close to the neutral pH, so that cytosolic proteins can work at its best. Now this antiporter is also called band 3, or you can remember anion exchanger 1 in case of red blood cells. Now this antiporter also helps in the carbon dioxide transport by red blood cells, and how this antiporter not only maintain the pH of the cells, but in case of red blood cells, for the particular red blood cells, it actually helps in the transportation of carbon dioxide. How it occurs in my next video, I will also discuss. Thank you for watching, and always keep learning.